So bones are formed, basically there are cells called osteoblasts and osteoclasts, and they will use the nutrients, the carbon, the nitrogen, the sulfur from your food, basically from the amino acids that are cycling in your body. They will use them to build your bone, but they will also be breaking the bone down. It basically makes sure that we don't that we've got this constant kind of refreshment of the amino acids and the kind of the nutrients of our bones. So that's constantly happening. Our bones are constantly being flushed with blood plasma, so they're constantly being, broken down and rebuilt. And so that process is constantly happening in bones. It doesn't happen in teeth. And so that's what turnover is. It's basically this constant breaking down and replacement. And the speed of that depends on, as I said, the kind of the bone itself. So we think it's because of the amount of loading. Your femur needs to take a lot of load, whereas your ribs not so much. And so it kind of depends on that, and then the type of bone as well. So if you think about that spongy bone, there's a lot more surface area that comes into contact with the blood plasma than the kind of the dense part of the bone. So it's actually an area that is there's still a lot of research to be done to really understand it, but that's basically the process and the reason why we've got these different, what we call turnover rates, the kind of different speed at which bones are broken down and replenished. So this goes back to this radiocarbon, and I have to say I don't do this. So I always get asked about radiocarbon. I always think, oh, it's not what I do, but basically it's to do with the fact that this carbon-14, which is another isotope of carbon, because it's got 14 neutrons in the nucleus, so not 12 or 13 like some stable isotopes, it's not stable. So it loses a neutron and it decays. So if you know about half-life, it has a particular half-life, and so you can basically measure. So it's when someone is living, when any organism is living, plants, animals, humans, they're constantly, again, that carbon-14, that radiocarbon is constantly being incorporated into their bones. When that organism dies, that stops. And so it's basically the kind of clock is stopped. And then the carbon-14 that's still in the bones, that's currently in the bones, will start to radioactively decay. And so by measuring the amount of carbon-14 that's left in the bones, you can work out, you can kind of do lots of maths to work out, to extrapolate, well, how long has it been since that radiocarbon was being replenished? And the half-life, so you can basically date people using radiocarbon, or anything using radiocarbon, up to about 40,000 years old. After that, basically there's no more radiocarbon left, it's all decayed. But until that point, you're able to get a pretty good idea of how long that person, for example, has been dead. I didn't do the DNA sampling, but I'm not. I don't quite know the rationale for doing the DNA sampling from the child as opposed to the mother, because they essentially are telling the same things. I guess by looking at the child, you get the father's information as well. So you're getting the mother's information sort of for free, and then you're also getting the father's information. Because basically they tend to take DNA from the petrous bone, it's the ear bone, it's very, very dense, and you tend to get the best preservation of DNA from that. I'm not actually sure exactly what they sampled in this case, but they likely took it from the unborn child, one of their bones, and then they got, that means that we can get information about the ancestry, or kind of of the mother as well as the child and the child's father. Carbon isotope values are basically all negative, and it's really annoying. It's because so we measure isotope, the relative amounts of the heavy and light isotopes relative to standards. So basically about 50, 60 years ago, when this first field was kind of discovered, people chose some what we call standards, reference materials. so that we could compare absolutely everything else to them. And they chose, for the standard for carbon, a fossil. It's a belemnite, it's a marine kind of fossil, and that has and they set the carbon isotope value of that fossil at 0. And they did the same for nitrogen, and that's actually atmospheric air. But for carbon, that fossil actually has a lot of the heavy isotope compared to absolutely everything else, which means that anything which has relatively less of the heavy isotope compared to that fossil has to have a negative carbon isotope value. It does make it quite difficult to talk about because you end up with everything being negative, and sort of talking about more negative, less negative. But that's why carbon isotope values tend to be negative. It's just because they're relative to an arbitrary material that was chosen about 60 years ago as the kind of 0 point. And so that 0 point is different for each element. So for carbon, nitrogen, sulfur, oxygen.